Okay, so good morning. Welcome to our final BIO205 review. I just want to say congratulations on getting it this far. Um, I think everybody is still in the class, those that did not drop out early for other reasons. So congratulations, you're almost there. For some of you, this will be your last class before moving on to your next programs or your next degree or wherever it might be. So again, I just want to congratulate you for getting this far. It's been a very good semester and here to help you get to the finish line so you can move on and go do what you got to do. Um, so what I'm going to cover today for this one is I'll start with Unit 4. Typically, depending on the questions or discussions that we have, sometimes these go longer than the time Zoom lets me. So I want to start with Unit 4 since that's the newest one. For the other units, if we don't get to them, a lot of the content is similar to my unit reviews from the previous exams. So I just recommend to go to look at those if we do run out of time. Biggest thing for the exam, just remember, is it's going to occur next week during your normal exam time. So coming in 8:30 a.m., you'll have 2 hours to complete the exam. You can bring your own laptop or tablet, or if we don't have that, we do have laptops available for you. We'll be using Respondus Lockdown browser for this, and it's all multiple choice, so no short answer questions. You'll take the final exam, you'll have your final grade, and then you'll know what you got in the class. Dr. Cotter is finishing up the lab grade and is moving those over to the lecture course. So if you are curious what your current grade is in the class, just go to your lecture course, and that should have your summary of lecture and lab, and that's what's going to be submitted for your final grade of the class. Before we get to that, if you have any tutorials that are left over or that you did not maximize your points on, you can still do those up until this Sunday. So if you want to try to maximize those points and uh, bump up your score a little bit before the final exam, I recommend you doing those. Traditionally, we do drop the three lowest tutorials. Um, I've not seen her do anything other than that, so I do expect that the three lowest tutorials will be dropped. But if you're, looking for some easy points or trying to give yourself some buffer before the final, I recommend you go ahead and do those. Okay, starting on Unit 4, this will also help you if you have not done your Chapter 13 case study, which is due tonight at 11:30 p.m. Okay, so the biggest thing with Unit 4 is primarily the infection chain. So the infection chain revolves around four main points. It's the illness or infection, the causative agent, which in our class is always a microbe, the reservoir, so where the microbe itself is residing, hiding, or living. Some examples of that could be contaminated food, stagnant water, dirt, soil, another human being, or another living organism. And the mode of transmission is how the microbe got from the reservoir to the host. So our case is built on um, a person getting infected, so that would be the host. The mode of transmission is how did that microbe get into the host itself. Some examples of that, a vehicle transmission, that is food primarily. Vector transmission, that's mosquitoes, fleas, ticks. Indirect contact is a non-living thing that was touched that got the microbe to jump from that non-living thing to the host. And then direct contact is typically um, living organism to living organism for the most part. An example like that is like kissing or coughing next to a person, being around someone, that kind of thing. So I have a couple examples down here. We do have some examples on page 1 of your study guide. I recommend that you have those completed because most likely one or two of those cases will be on the final exam. But example, an illness such as strep throat, causative agent is Strep pyogenes. The reservoir for strep throat is typically a human. So humans will transmit those airborne droplets, and then other people around them will get sick, and then they'll develop a strep throat infection. And then things like botulism caused by Clostridium botulinum. Give me 1 second. I'm going to mute someone. Um, there we go. Perfect. Um, yeah, so consider botulinum again, your cause of ancient, that's your microbe reservoir. In this case, it's typically contaminated food for honey, and then your mode of transmission is vehicle, which is you ate the contaminated honey. Okay, stages of infectious illness. So this is kind of, I'll use myself as an example because I just went through this recently. So there's 5 stages of illness. And basically, the way it begins is from the time that the microbe invades your body or the host, and then working its way all the way until you start the recovery process, and the body is able to begin repairing its cells, its organs, etc. So starting with inoculation, typically this is where the microbe infects our tissues or our cells and starts replicating. For the most part, inoculation, you don't really see symptoms. Things like what I had was the cold. I could be have that inoculation period go for about 3 to 5 days with no symptoms. Other things like COVID, if you recall, those would have longer inoculation times, about a week to 2 weeks before the patient started seeing illnesses. And then some other infections, things like the flu, inoculation is very quick, maybe a day to 2 days before the flu virus starts actually replicating enough for the body to start showing illnesses. Um, as we work through inoculation and go into the prodromal symptom, this is usually the first sign of someone getting sick. You know this because you sometimes feel weak or lethargic. You might feel lightheaded. In my case, my first symptom was a very rapid sore throat that I did not have before, which is typically the first sign that you're actually getting a cold infection because it's a respiratory infection. Um, but you'll have mild symptoms. You'll start feeling sick before you're actually sick. And then the active illness, this is where you start having the fever, the chills, coughing, sneezing, um, all the signs and symptoms of when you get infected. This is typically the most severe stage of the illness. So right now, the infection is trying to um, take over and beat out your immune system. So this is typically where the immune system is trying to play catch up and trying to control the infection. And like we talked about in Unit 3, the immune system is able to learn how to beat almost every illness. The problem is, do you have enough time before the infection causes so much damage that your immune system is not able to actually beat the infection in time? That's kind of what happens here is this is a point where you might have death or you might actually have to go in for medical treatment because your body by itself cannot um, defend itself fast enough. Once you've actually started to control the infection, you'll start getting a decline period. This is where you start feeling a little better. Maybe the major symptoms are gone. We might still have some of the minor symptoms. And then convalescence is your body has defeated the infection, and now the body is using its energy resources to start repairing your cells, your tissues, that kind of thing. Quickly, endotoxins versus exotoxins. This is in your note in your notes, but you might get asked questions about the difference between an exotoxin and an endotoxin. One of these might be on the case study if you haven't done it already, and just kind of biggest things to tell them apart. Which is in a gram negative versus gram positive, which is a lipid versus a protein, which are heat stable, and then primarily which one causes the local or systemic effects. Um, Expect maybe like a question or two on this on the exam. I wouldn't burn too many calories looking at this. Just being able to compare and contrast the two. Um, Maybe give 3 to 5 of these and you should be good to go. I wouldn't worry about this one too much. And then types of exotoxins. You might get asked a question for what are the 5 exotoxins and give an example. And you can kind of see here, I wrote this out for you. Feel free to take a snapshot now or later, but this should be in your notes as well. Some of the stuff I might power through real quickly, just because I think it's not as important as some of the other stuff I'll cover later. And then virulence factors. This is in handout pages 10 to 16, kind of just discussing the factors for virulence. How do infections help infect us? So things like attachment. Remember, we have pili and spikes. We also have things that help microbes avoid our immune system, like the glycocalyx, certain enzymes, also their antibody receptors. And then there are also things that they can do to cause damage in our body to make it easier for them to invade. Things like releasing toxins, like those endotoxin we talked about, or direct damage where they actually will go and destroy our cells to slow down our body's ability to fight them off. And then just a quick example of what endemic outbreaks, epidemics, and pandemics are. Again, if you might get a question or two on these, but I wouldn't burn too many calories. Most of these definitions are fairly straightforward. Okay, so going back to Unit 1, I know this was a long time ago. So very quickly, Koch's postulates. I do expect a question on this one. I haven't seen the final exam, and sometimes she rotates questions in and out. But Koch's postulates is always one that gets missed, like I mentioned in Unit 1, and especially now, 15 weeks into this gets forgotten. Just remember that Koch's postulates, we're going to observe the patient for signs of illness and isolate the host or the microbe. And remember that signs are what can be seen, symptoms are what the patient feels, and being able to just list out which are signs and symptoms in your case studies is going to be very helpful. Next, we're going to culture and identify the microbe in the lab using different techniques. Just like before, pick two of your favorite techniques, microscopic, macroscopic, biochemical, and just be able to briefly describe what those are. Three, infect the new host with the same microbe and observe for the same signs of illness. Um, and then four is the one that I see get missed the most is remember that we have to use the same test in step 4 that we do in step 2. Don't change up the test because then you're gonna get that question wrong. And I'm gonna look at the chat real quick. Uh, yeah, yeah, Cindy, good question. So the question is, have I seen the exam? I have not seen the exam. I typically don't see the exams until the day of or basically after you guys take them. So this is a combination of all the exams I've seen over the last 4 years. I'm kind of picking things that I've seen. As a TA, I don't get access to see the exam until she posts it for me. And typically she doesn't do that until the day of. Does that kind of answer your question? Did you have anything specific about that you want to talk about? If not, I'll keep going. You can just throw it in the chat. But for the most part, I have not seen this specific exam, but I have seen all of the other exams we've taken in other years, and um, some of the questions are very similar, and then she might change up a couple or so. Okay, so your question is on the case studies. Um, Realistically, remember how, each of the unit case studies were maybe like 2 to 3 cases? I would expect about 5 to 6 cases on the final. What might be different about the final, though, is that let's say it's a case on like unit 1. So the case for unit 1 might ask you to, hey, describe Koch's postulates and give me the signs, give me the symptoms, that kind of thing. Um, but then thrown into that question for another part, they might ask you like, hey, you just saw viral spikes, hemagglutinin. What are the factors of replication for the virus that has hemagglutinin? And you're saying, oh, hemagglutinin is influenza, so it has these replication steps. Um, I'm making it more difficult than it actually sounds, but again, these are all multiple choice questions. So there's no like, oh, I've got to type out all this stuff. It's really multiple choice questions. It's just now that where it's a combined exam, you might get pulled a question from Unit 2, a question from Unit 3. A very awesome way or very good way to think about this is if I give you a microbe, I might ask you, hey, what's a physical method you could control this microbe? What's a chemical method you can control this microbe? What does your immune system say? Dr. Cotter stated on Wednesday. Oh, interesting. Okay, so see, that's new. She might have added that write-in portion. Before this was all multiple choice, Angelica. So I do appreciate you mentioning that. I did miss class that day. So see, it looks like she did add a write-in portion. Um, so yeah, so it sounds like we'll have some writing portions. But in my experience, it's Well, I guess every test I've had before this has been a multiple choice test. So I would expect if it is a write-in, it may not be a lot of write-in questions because we do try to get the grades out. We have to get the grades out by Thursday or Friday, I believe. And so we don't put, she probably won't put too many multiple written answer questions because then she has to go and grade all of those. So if she does add some of those, I'd expect a few of them, maybe not too many though. Thanks for bringing that up, Angelica. That's a good point. And then, okay, sounds like I answered your question, Cindy. And if you have any other questions, just message me after this. We can always go over that. Again, cell parts, like I mentioned in Unit 1, cell parts are important to know, not because of what they do, but how you can use cell parts to identify different organisms. So again, bacterial cells or fungal cells, they both have cell walls. But again, bacterial is typically peptidoglycan, whereas fungal cells have chitin. And so being able to compare and contrast those is going to be helpful in the exam. Same thing with the viruses. We talked about the viral spikes, hemoglobin, neuraminidase, and GP120, GP41. There's no other virus we ever bring up in this exam. It's always going to be HIV or influenza. Um, and again, just kind of go back to Unit 1 and just remember which parts tell you which type of organism it is what I would really focus on. Kind of same thing here. Cell shapes. Again, you might get asked a few questions on this. But I would really just briefly review this, then move on. I wouldn't burn too many calories on the cell shapes, but you have it here in case you want to review it. And like I mentioned, the big thing is, like we've been talking about in Unit 1, 2, 3, and 4, is can you compare and contrast the microbes based on the information we give you? The gram stain, does it stain pink or purple? Does it have a cell wall? Does it have chitin? That kind of thing. Um, as long as you can break those apart, you should be okay. And then the viral infection replication chain, like I mentioned before, you have the six steps: attachment, entry, uncoating, replication, assembly, and release. And then being able to identify the differences between the two viruses we talked about in this class. So HIV will have the GP120 and GP41 viral spikes. It infects the CD4 helper T cells, and it utilizes the enzyme reverse transcriptase to replicate. Whereas influenza uses hemagglutinin and neuraminidase. It infects the respiratory epithelial cells, and it uses straight RNA polymerase, so no going backwards into DNA, then back into RNA. Sorry, I'm a little flummy right now. Give me a second. Okay, so Unit 2. I got rid of some of the slides that I don't think you need really to focus on that we're really talking about like mesophiles, thermophiles, that kind of thing. Again, briefly look that over. It's not too complicated, but I don't want you to burn calories on that. The things that you will have to focus on, horizontal gene transfer. So remember that we have um, three different ways that bacteria can transfer genes. And what it means by horizontal gene transfer means that they're not replicating to transfer these genes. They're able to exchange genes just by being next to each other. So you have conjugation where you have the sex pilus, and essentially some of the DNA from the plasmid gets moved over into another cell, and then that other bacterial cell now develops the same antibiotic resistances that the original bacteria had. Bacterial transformation is typically when dead cell fragments are exposed, and a live cell is then able to eat those fragments up and incorporate that DNA into itself, and they're able to then take on certain characteristics and antibiotic properties of the dead cell. And then transduction, especially for the Wednesday group, you saw this a lot with phages, where a phage will try to infect its viral DNA. It accidentally will take a piece of the bacterial DNA, and then it'll infect another bacteria with that bacterial DNA. And now that second bacteria develops new properties that it stole from the first DNA or the first bacteria. Again, drift versus shift. Being able to compare and contrast the two. Again, it's not going to be many questions on this, maybe like one. Just understanding that drift is a small change caused by point mutations, and then a shift is typically created by a major change in the viral spike structure, and it's due to genetic recombination, like shown here. Okay, the stuff that we do need to focus on is methods of control. Like I mentioned before, I can ask you a question about give me two ways to sterilize, physical ways to sterilize this microbe. Give me two ways to disinfect this microbe. Give me two ways to chemically sterilize, chemically disinfect. Um, I kind of have it broken up like this, where okay, what are my physical sterilants? Autoclaving, ultraviolet light, and filtration or mechanical filtration. And my chemical sterilants are the Oz, aldehydes, halogens, and hydrogen peroxide. And then disinfectants, kind of listed here. This is good for note cards if you ever did note cards for Unit 2, or if you have like a Quizlet or something like that. But I think the biggest thing to remember is, you know your physical sterilants, know your chemical sterilants, know your disinfectants for physical, disinfectants for chemical. And then on the exam, we'll probably get asked, hey, list one or two of each of these or something like that, or select physical sterilants, and you'll select from a multiple choice group or something like that. And then the antibiotics, page 10 of the study guide for Unit 2. Um, know what drugs target what. So plasma membranes, there's only one antibiotic that targets the plasma membrane. We've got a couple that target the peptidoglycan cell walls. We have a few that target the waxy cell walls and folic acid metabolism. The big thing with this one, remember which is to note which one is more or less toxic, because that could be asked on the exam, and that should be in your page 10 of the study guide. Protein synthesis, target drugs, and also which is more or less toxic, and then which are your DNA and your antiviral. Um, Again, I think more of the questions for this might be asking you, how have I seen this broken up? I've seen this broken up as a question where it basically says, hey, you've diagnosed it to this Staph aureus with a waxy cell wall or something. Um, what is a drug that can target the cell wall? What is a drug that can target DNA? And you just select from a multiple choice list. You might not have too many specifically to this one, but I'll just go over page 10 and just be able to group it like the cillins. I don't care which cillin you use, penicillin, ampicillin. Those are probably all going to target the same thing, so just pick a few of those. Ooh, okay, Unit 3. This is the one that we just did. I didn't look at the exam scores on this one, so I do apologize for that. But again, going back to the infection chain, say that I have a flu. Um, what are the physical barriers that would have protected me or that were trying to protect me during this flu infection? Cold infection. And it was a respiratory infection. So I had a lot of mucus built up. I had a lot of coughing, a lot of sneezing. My mucous membranes were constantly secreting mucus. And then obviously the hair in my cilia in my nose was trying to catch it and then deflect it all off. So knowing your physical barriers, again, I would focus on respiratory, GI, and reproductive, because those are very common to one another. They mostly have mucous membranes. They all have mucus. And you have the small specific ones where if it's an E. coli infection or stomach infection, I'm probably going to vomit or have diarrhea, um, versus respiratory where I have the coughing and the sneezing. And then the chemical barriers trying to protect me during my cold infection was lysozymes and lactoferrins specifically. But you can kind of see where like the GI tract has a couple extra things. You have the mucus and saliva. You also have hydrochloric acid, bile and enzymes, and gastroferrins. And then reproductive also has mucus, but it's also very acidic in the reproductive system. And you also have those same lactoferrins. But again, the way I expect this question is very similar to Unit 3, where it gives you the case study and tells you give me uh, two physical barriers for first line, give me two chemical barriers for first line, and then you might get asked a question about the resident microbes and why we have them. Um, Take an example. Against we've played a lot with Staph aureus, Staph epidermidis in class. We've played it with E. coli in the GI tract, and then for the vagina, typically Candida albicans is a very common one that she sometimes asks. So if it feels like, hey, this is a an infection in the vagina, this probably is your candidate. Um, just throwing that out there if you see it. And then second line defense, the same questions are probably going to be asked. What two second line cells are involved in every immune response? Give me some of the chemicals that they use and then the processes that they use to help us fight it off. Um, and so this is from page 14 of your Unit 3 handout. I think for the most part, y'all did fairly well on this. But the way, especially Unit 3 is a little easier because we just had it. And some of those questions will probably get repeated on on the final exam. And then third line, I think this is still where people struggled, from what I remember talking with Dr. Cotter about, is being able to differentiate your two third line cells. So if the infection is, I'm just going to say, if it's a viral infection, it's going to be an intracellular response, or what they call it's intracellular, or they'll call it um, a cellular response or an intracellular response. Just know that if that is involved, you use Th1 helper T cells using the CD4 receptor. You'll have CD8 cytotoxic T cells and memory T cells. And then the T cells will use the T cell receptors to screen other cells. And then the cytotoxic T cells will destroy the infected cells using perforins and granzymes. If the infection is anything else, allergies, bacteria, fungus, parasite, um, it is an extracellular response, or what you might read as a humoral response. Humors meaning like blood in this case. You're going to have the Th2, CD4 plus helper T cells, again using that same CD4 plus receptor. It's going to activate plasma B cells and memory B cells. It will use the B cell receptor to look for invading microbes. And then B cells do not kill, they secrete antibodies and surround the microbe to agglutinate, neutralize, optimize, and perform complement fixation. Agglutinate makes them all the antibodies will attach to the microbes and then they'll attach together like Legos. So it kind of clumps them together for our macrophages to find more easily. Neutralizing, the antibodies will block off the sites of attachment on the bacteria. I'll use bacteria in this example, and that prevents the bacteria from actually attaching to our cells, our lining, so it can actually try to infect us. Optimization, the antibodies will secrete chemicals that make it much more appealing for the macrophage to want to digest. And then antibodies can also perform complement fixation. Remember the C5 through C9 MAC attack? Antibodies basically help set up a site for those complement proteins to line up and start performing the MAC attack. And then knowing your antibody types, remember that when I have an infection, I'll start with the IgM, which is typically the biggest one. I'll then go into IgE, which goes anywhere, including the placenta, can pass that barrier. If I have any kind of mucus infection or respiratory infection, I have IgA involved. And then if I have any kind of allergies, I will be using IgE instead of IgG. That's as easiest as I can describe it for you guys. Hopefully that helps on the exam. Okay, since I got the notice that I only have about 7 minutes left, just my final tips. The study guides are going to be your friend on this final exam. Anything on the unit study guides can be on the exam, so use them as your case study. Um, I know I put here it's multiple choice. For the multiple choice ones, if you're not sure on an answer, just get rid of the ones you don't know, and then give yourself a 50-50 chance to guess the right one. Another thing is you're not penalized. You don't have to answer the question right away. If you don't know the answer, skip it, move on to the next question, and then just keep working your way and then work your way back to the questions you missed. Um, Again, you do have 2 hours. I've typically seen, I'd say half the students finish within the first hour, hour and 15 minutes, and the other half will take up to the 2 hours. But I have seen some students not finish the exam because they kind of stayed on a question for too long. So don't do that to yourself. Give yourself a chance to answer the most questions possible. And if you're running short on time, and if it's multiple choice, just start selecting stuff. You're more likely to get that right than not answering and getting a guaranteed zero. Um, this one doesn't apply. This is more for the hype for our online group, but we'll be in the lab. Bring your laptops, bring your tablets, make sure Respondus is set up. And if you do have any concerns about your laptop or your tablet, just use our computer. Um, I don't want you to get kicked off halfway through trying to get the exam to work. And then just in general for your all your finals, make sure you're getting a good night's sleep. Make sure you eat something, and then bring your coffee, bring your tea. You can drink that in the lab. And then I'll have something to give you all as you walk out. I did promise I'd make something for you all, so I will abide by that and bring something in so you can walk out and snack on something. That's all I had for this review. Does anyone have anything they want me to go over in the next last 5 minutes that I have before I close this up? You can raise your hand, you can unmute, or you can throw it in the chat. Okay. And thanks, Aniston. I appreciate it. Good luck on your finals. If no one has anything, then I'm going to go ahead and give you back your 5 minutes. And if anything does come up between now and then that you have a question on, feel free to reach out to me. I'll be available. And I'll be on campus early on Monday and Wednesday to kind of talk anything over with you all as well if you need it. But okay, I'll go ahead and stop recording and I'll let you all go. Have a good weekend.